people have a misconception that insects tend to be scary and dirty. Many of them are extremely small, and those bigger ones, they don't look aesthetically pleasing. So for example, a centipede or a millipede. First instinct is usually to get scared of them, and they might even try to kill them. But actually they are a very important part of our ecosystem. I'm Li Si, a Year 4 undergraduate at the Asian School of Environment, majoring in environment science and specializing in ecology. My project focuses on soil and leaf litter arthropods, or invertebrates, in our forest in Singapore. This project is part of the Uriga program. So Uriga stands for Undergraduate Research Experience on Campus. So it's basically a research experience designed to allow students to take part in a research project that is 1 year long, so it gives them insight into the world of research. For my project, the goal was to sample the soil and leaf litter arthropod diversity and community across different forest types in Singapore to answer this question of whether forest restoration is actually helpful in restoring the diversity and community. In general, arthropods refer to this group of cold-blooded animals without backbones. And within this phylum, there are 4 different subphylums. You have your myriapods, chelicerates, your crustaceans, and then the last group are hexapods. We sample across 4 different forest types. This includes your typical primary forest, secondary forest, restored and unrestored forest. The first thing we do is to set up our quadrants. The way our quadrant works is basically a square. So each corner of the square is 10 meters apart, and then we'll sample the soil and leaf litter from these 4 corners. At the end, we'll also sample right in the middle of this square. So in total, we'll have 5 different sampling points. After we designate the 5 different points, the first thing we'll do is to collect the leaf litter. So to collect the leaf litter, we use this contraption called the litter reducer. So essentially it works like a sieve in which you put all the leaf litter in and then you shake it, and then the smaller leaf litter parts as well as the invertebrates within, they'll fall out and then they'll be captured, and we'll transfer them into a Ziploc bag. After that, we also sample the soil. So for this part, we'll basically collect all the root mat / humus kind of layer, and then we'll dig it up and then we'll transfer them into the Ziploc. And after everything, we'll bring all of them back to the lab to extract the invertebrates and arthropods within. Our samples will go through 2 different extraction methods. So the first one is the Belize Talgren funnel. It's basically like a bucket. We'll transfer all our soil samples into the funnel itself, and we'll cover the top with a lid that has a light bulb inside. The heat, as well as the light from the light bulb, will dry out the soil samples, and because the arthropods within, they are sensitive to light and heat, they'll move downwards and they'll drop into a bottle of ethanol where we'll collect the arthropods. In the other method, it's called Winkler extractor. So the Winkler extractor contains smaller mesh bags where we'll transfer all our leaf litter samples into, and at the bottom of the Winkler extractor there is a bottle of ethanol. Over the course of 1 week, mesh bags starts drying out. You'll start to look for places with moisture, and by random chance and motion, these invertebrates will drop out of the mesh bags and drop down into the bottle of ethanol below. And that's where we'll collect the ethanol samples and bring it back to the lab for identification. After we bring the specimens back into the lab, we will identify them up to their order level. So to do this, we'll have to look through the specimens that we have under a microscope, and then we'll make use of identification keys to identify them to specific orders. So for orders, this includes things like centipedes, millipedes, ants, termites, cockroaches, grasshoppers, that kind of thing. And after we identify them to their order level, we'll collect the specimens and we'll set up a reference collection. And after that, we also collated the data which was used for analysis for my project. For our main findings, we found that forest restoration in heavily degraded forest sites does indeed help to restore the leaf litter and arthropod communities, albeit at a very slow rate. This means that even though there has been a long period of restoration, the arthropod communities aren't that much different from its original state. Another interesting finding that we found was that we should also focus our forest restoration efforts on the mature secondary forest into their primary forest stage. This is because the communities in primary forest tend to be distinctly different from the other communities in the other forest types, namely the secondary, restored and unrestored forest. So they are an incredibly diverse group of animals. In fact, there's over 1.2 million species discovered to date, and amongst all the animal species, this is about 80%. They perform a lot of important functions in the ecosystem. So in our forest, they do important jobs such as nutrient cycling, improving the soil quality. They also act as food for predators. They are also a big part of the food chain. So there is this saying that I included in my research paper, which states that even though these arthropods, they do not have a backbone, they are undeniably one of the most important backbones of our ecosystem because they perform such important functions. Insects in general, they are quite a misunderstood group of creatures. One thing you can think of is that actually they are as afraid of you as you are of them, and their first instinct is not to bite or hurt you, so you can just keep your distance and it will be fine. Yeah, and you might even get a chance to observe some interesting behavior.